Okay, so this is starting Unit 2. Unit 2 deals with the erythrocyte, hemoglobin, and then we're going to talk about the CBC in general. Okay, the complete blood count, what you do when you do a CBC, talk about a diff, talk about morphology, those kind of things. So starting pretty soon, your labs are going to get a little harder because you'll be responsible for doing a diff and a morphology on the slides. So it will take a little bit longer, but it's not that hard. I mean, you know, it just will be more time consuming as we go. So today we're going to start the erythrocyte. I'll try to get the study guide up online for Unit 2 today, this afternoon, so you guys will have it to go ahead and start studying. Look like you guys was clicking through pretty quick on the study guide. Now listen, I appreciate that, and I hope you do really, really good, but you don't need to memorize this stuff, okay? This is stuff you need to learn, okay? So try not to memorize the study guide so you can make 100 on the test. If you're not learning the material, it's not going to do you a bit of freaking good, okay? Because you still got to pass your boards, right? We got to pass boards. That's the important thing for this class and this program, okay? All right, so this is chapter 5. So let's talk about the erythrocyte in general as we go. All right, so erythrocytes, of course, are the red blood cells. Their main job is to carry oxygen, okay? They do carry CO2, but that's not their main function that we normally think about. Their main function is to carry oxygen to be able to do glycolysis in the tissue, okay? A little later on, we're going to talk about glycolysis in the red blood cell, and it's actually done without oxygen, okay? Because most of the red cells don't have as much oxygen, or the oxygen they do have, they give it up to go to the other tissues. So inside the cell itself, it doesn't have oxygen available to do glycolysis. So it actually does a form of fermentation, anaerobic glycolysis, that we'll talk about a little later on. Okay. The word anemia. The word anemia means an insufficient number of RBCs. So patients who have low RBC counts are known as anemia, okay? Now, there are some anemias, and let me go ahead and say this, that they have a normal RBC count, but their hemoglobin is low, okay? Or it's a non-functioning hemoglobin. We'll talk about that later on. So a lot of times anemias are not only a decrease in RBCs, but it's also a decrease in hemoglobin content. And so that's one of the things that a lot of people get confused about, because sometimes you can have the red cells, a normal red cell count, but you just don't have the hemoglobin in them. And we refer to those patients as anemics also. Okay? All right. Questions about anything so far? Okay. New suffix, cytosis. Anytime you see the word cytosis, it means an increase in cells. Okay? So in this case, if we have erythrocytosis, that means we have an increase in RBCs, all right? So normal RBC count, and I'm just going to put a big number up there, is anywhere, I'm going to round it off, is anywhere from like 4 to 6 times 10 to the 6, which is million per millimeter cubed, or a microliter, okay? That's a good RBC count that would cover males and females, okay? Now, if you'll open up the front of your book where those normal ranges are, you're going to see a little bit different. Like females is like 4 to about 5, whereas males are like 5 to 6, okay? So just in general, if we say 4 to 6 million per millimeter cubed, we're covering both male and female, okay? So erythrocytosis would be anything greater than like 6. So if you had a patient that had like a 7 or an 8 million RBC count, then they definitely have erythrocytosis, okay? We'll talk about later on, there is a hereditary disorder that's called hereditary hemochromatosis, and they have erythrocytosis. Hemochromatosis. I think is how it's spelled. Hemochromatosis is a genetic disorder. It's not really anything to do with the red blood cell. It's an iron thing. Okay, so like Normally, us normal individuals that don't have hemochromatosis, we lose about 1% of our red cells a day. Okay, so when we lose those red cells, when we take in our dietary iron, we absorb just what we lost. Does that make sense? We don't get extra and store it in. Our body's not designed that way. So, but people that have hemochromatosis, their body has lost that ability to know how much iron to store or how much iron to take in. And so they take in all of their iron. Every time they eat something that has iron in it, their body absorbs it and puts it into circulation. Yes, ma'am.
Why does it make you like sick if you if your iron is normal and you like take iron? Why does it make you like sick?
Like how sick?
Well, like I used to be after I gave birth and my iron was low, so I had iron in my birth control.
Mm-hmm.
But then, like if I forgot to take it one night and I took like 2 pills the next day, like I looked and felt like I was dead.
Make you like nauseated and that kind of stuff.
Couldn't stand up.
It's just because your body just didn't know what to do with it.
Okay.
And that's usually what happens. If you can actually get iron toxicity. Too much iron becomes poisonous to you in your body. And we're going to talk about iron in the chapter with hemoglobin.
Mm-hmm.
So like most of the iron that we have. So remember from chemistry, have all you guys had chemistry yet?
In it.
Or in it right now? Okay. So iron, if you look on the periodic table is shaped like this, right? So from about here, and I'm just making this up as I go, okay? All of that from that side over is metals, okay? Then you have a group of metals that fall right here. So you've got like 1A, 2A, and then you got 3 through 8A. Here, over here on this side. But this group is known as your B group. And they're known as that because these guys pretty much stay in a certain order with their valence shell, So like if they're metals, they'll always be, like these will all be +1. These guys will be +2, 3, 4, and then when you get to 5, they're -3 and so forth as you go. These guys don't do that. So they like vary in their charge, okay? Iron is one of those. Iron can be in the form of +2 or +3, okay? Most of the iron that we, hang on. Is that right? 3? Yeah, I know, but +2, +3. Yeah, +2, +3. And, well, no, that wouldn't help me, but I appreciate it. But there's a ferrous and a ferric. Okay? In our body, we want the +2. That's the only way iron can be used in hemoglobin. It has to be in this +2 form. And so we do an oxidation process to take iron from +3 to +2. Okay? Most of the iron that we take into our body is in the +3 state. And so it has to do that. So if you take in tons and tons of extra iron, it's bringing it in that +3 state. So therefore your body has to do lots of oxidative processes to make it to +2 so it can be used. So therefore it causes problems to your body. Okay? Another thing that, like what we were talking about here, and with hemochromatosis, this is their problem, is they don't have that ability to stop absorbing iron. So they absorb iron all the time. So their body gets it, absorbs it, and then changes it to F2. Well, your body's very, very efficient. It says, hey, we got all this extra iron, we can't just have it hanging around, so we're just going to make red blood cells. Because that's what iron's used for, is for hemoglobin production. If we make a bunch of hemoglobin, then we need something to put it in. And so our body begins to do erythrocytosis, and we end up having way more red blood cells than we need. And so the therapy for hemochromatosis is basically phlebotomies. We do therapeutic phlebotomies. We draw blood off of them, okay? A unit off, all right? Okay? So erythrocytosis, excessive number has no adverse effect on pulmonary gas exchange. So what that means is, just because you have tons of red blood cells doesn't mean you're going to become hypoxic and, overload of CO2 because we're sucking all the oxygen or putting oxygen out and those kind of things. It doesn't change. Because, here's why it doesn't change, is because oxygen and CO2 exchange is based off of your tissue activity. It has nothing to do with your red cells. Your red cells are just a means of transport of oxygen and CO2. Does that make sense? So it doesn't matter if you have, 10 trillion or 10 billion red cells, it's still, your body's still going to do gas exchange exactly the same way. Does that make sense? Okay. All right. Okay. All right. Erythron. Okay. Erythron is a new term you guys probably never heard before. Erythron just essentially is a term, general term used to describe all of the red cells in your body, from the most immature form in the bone marrow to the circulating mature form. If we're talking about the whole red cell population, we're talking about an erythron. Okay? All right? So the totality of all the stages of the erythrocyte. Erythron. All right? Erythropoiesis. All right? So we know what hematopoiesis means. Hematopoiesis is what we've been talking about in the last unit. Erythropoiesis is just concentrating on the red cell production. Okay, so basically from that HSC to the mature red cell that you see on your slides. Okay, that's what erythropoiesis is. So that's what we're going to spend some time on in this chapter. All right, so it is an orderly process that should always happen in an order. Okay, we're going to talk about that order today when we get a little further in. But essentially you have three types of cells, or I'm sorry, not three, but what we're going to talk about. We have HSC. What is HSC?
Hematopoietic stem cells.
That a girl. All right. Then the next one, this group basically matures into what we call B. All right, this is burst. It stands for burst.
Forming unit, you referred?
Yes, forming unit erythroid. Okay, then the next maturation is CFU. E, which stands for colony forming unit erythroid. Okay, now I kind of lied. Here you have CMP. Remember that common myeloid progenitor. Okay. But once this guy has been exposed to like EPO, and I think it's interleukin 3, there's a couple of other ones. Once they're exposed to those guys, then it forms this erythroid commitment. Remember how we talked about commitment and it's going to become that one unilineage, okay? I used that term a couple of times last unit. Unilineage means it's just going to go to one cell. And of course, in this case, we're talking about erythroid. Okay? Then, it is going to be the rubroblast. Okay? Now, this guy's the first recognizable. I can't spell recognizable. How do you spell recognizable? It's got a G in there somewhere, doesn't it?
R-E-C-O-G.
O-G-N-I-S-E. Recognizable.
Is it Z or?
Yeah, I think it's a Z, isn't it?
Okay.
Looks close. You know what I mean? Okay? So in other words, here up, we don't recognize what they are because they all kind of look the same. They look like those little lymphocytes. Remember how we talked about that? But once we get to the rugoblast stage, okay, rugoblast is also called a pronormoblast, isn't it?
Mm-hmm.
Okay. So the pro normal blast, and then of course it'll go to the pro Ruber blast. And this one, is this the basophilic normal blast? Is that the next group?
Yeah.
Or that new word? Basophilic. Oops. And hey, listen, all this is going to be on the slide in just a little bit, okay? I'm just getting ahead of myself. And then the next one would be the, I can't write this low. Erythrocyte. And then the next one is the Metarube. And then of course you got the reticulocyte. And I'm just going to call it retic. And then you got the RBC, the erythrocyte. Okay? And that's kind of the lineage that we looked at. So all of these are based off of the maturation is based off of the hormone erythropoietin. Okay? EPO. This one's very, very important. Yes, ma'am.
This is the erythropoiesis.
Yes, exactly. This is erythropoiesis. Okay? So EPO is the major regulator of erythropoiesis. Okay? And what's really unusual, all right, now these guys over here that we were just talking about, these have to do, we know they occur in the body, but we don't understand them fully as far as how they act in the bone marrow. These two terms come from cell culture. Remember I told you guys that last unit? Talked about how some of this stuff they found out because of cell cultures growing them in a lab, and that's how they acted. And that's usually why, that's where these two actually, we know they do this based off of cell culture in a lab, okay? But this, from here down, of course, we know is happening because we see it in the bone marrow. We can recognize these cells and their maturations, okay? All right? But, for instance, this one, the colony forming unit, actually responds to low levels of EPO, okay? So even at low levels of EPO, you can get colony forming units, erythroid cells, okay? They'll form, do what they're supposed to do, and then go into maturation for the rest of the thing. Whereas these guys, they don't form unless there's high levels of EPO, okay? So like sometimes you may not even see this formation in the bone marrow, okay? Because if EPO levels don't get to a certain level, they don't form, okay? So it's almost like it kind of skips and goes straight to the CFUs, okay? Does that make sense? Does that make sense? Okay? Because these two are basically, they form based off of EPO levels. Okay? So like if a patient has really, really bad anemia, okay, has a low red blood cell count or trauma where they're losing blood all of a sudden, their body is going to stimulate their kidneys to produce more EPO, erythropoietin. So then you're going to have this burst form units that will form too, okay? And that's essentially what it is. It's kind of doubling your immature cells to be able to get more of these, okay? Because these guys will actually do mitosis more than these guys do, okay? And it's all based off of the amount of erythropoietin.
Shouldn't it, I don't know, it doesn't make sense to me. Shouldn't they be like side by side? Do they happen at the same time?
They can, yeah. I mean, it's just a little flow chart, but yeah, I mean, yeah, they can happen at the same time depending on what's going on in the body.
Okay.
Yeah, I'm just doing like a progression because that's the order they kind of go in. All right, circulating lifespan, very, very important here. We say 120 days, always. This says 100 to 120 days. In your brain, I want you to know 120 days, okay? Because that's what everybody teaches, all right? And that's what you'll see on your boards. Circulating lifespan. So circulating lifespan, what does that mean? When it gets in what? The peripheral blood. Anytime, remember I told you guys, anytime we see circulating, we're talking about in the peripheral blood. Okay? Really what happens is that stem cell, when it's reproducing, you know, making or commitment to erythroids, and you form those BFUs or the CFUs, then those only take a couple of days to do their job, to get mature, and then get kicked out of circulation. So really you would probably be looking at, if you looked at the entire lifespan of the erythron, then it would be, you know, about 125 days, 130 days at the most. Does that make sense? But when we talk about 120 days, we're talking about in circulation. So once it's got the nucleus kicked out and it enters the bloodstream, the peripheral blood, then we look at about 120 days. Okay? Now remember what I told you guys. Every 120 days, you don't lose all your red cells, right? We only lose about 1% a day. Okay, and so it's kind of staggered so that we don't ever have any trouble with our blood. Okay, you know what senescent means? Senescent means old. I'm senescent. I don't work as well anymore as I used to. Okay, so these are like older cells that are inefficient. All right, so then they're destroyed in the liver, the spleen, and the bone marrow also by macrophages. And we'll talk about that. We talked a little bit about that the other day when I was talking about the spleen. And the spleen does a really, really good job. And it's our main one. But the liver also plays a role in destroying senescent cells. Okay? Questions about that, guys? All right. So, precursor cells in the bone marrow that are morphologically identifiable, we refer to them as erythroblast. Okay, no matter where they're at. All right, so from the rugoblast down to the metaroot, you will refer to it as a erythroblast. Okay, all of them are referred to as erythroblast. Okay, if they're normal. Okay, so in other words, the sequence is normal, their size is normal, everything is normal about their production, then we would refer to them as normoblasts. Okay, that's just another term that we'll use. So sometimes when we're talking and in lecture, looking in the book, it may say erythroblast. When it says erythroblast, it's not talking about any particular one, it's just talking about any of those that are nucleated. Does that make sense to you guys? Okay. Good information to know. All right. So later on, we'll talk about when we talk about normal. All right. When we talk about IDA, which is iron deficiency anemia, and then we talk about pernicious anemia, which is megaloblastic anemia. Okay. They don't proceed normally. Okay. So like the cells are smaller in IDA, they're known as a microcytic anemia. The cells are smaller because they don't have the iron to be able to make a normal size cell. So our body's efficient, so it just puts what it can in there to keep oxygen going. Whereas right the opposite of megaloblastic anemia, there's not enough. Well, I'm not gonna get into it, but the cells are larger. Okay, when we start talking about pernicious anemia, and I'll tell you why they are. When we get a little further in. But normoblast is if they're normal size, normal color, they proceed in the normal production. Okay? So erythroblast is just all enucleated cell precursors in the bone marrow. All right? Reticulocytes. Reticulocytes are also known as polychromatophilic RBCs. All right? Now, listen to me. You can normally see reticulocytes on a blood smear. Okay? The only way you'll know that they're reticulocytes, though, is they have a little blue tinge to them. Okay? Polychromasia is the word. Okay? Polychromasia. This is reticulocytes. Alright? Now, on if we're looking for real reticulocyte count, we have to use a supravital stain. You remember me using that term last unit? Supravital is like a bag of Skittles, okay? It is a stain that stains interior things inside the cell, all right? It can stain on the outside and the inside of the cell, okay?
What was it called?
It's called a supravital. Supravital stain. Okay? So this is the pack of Skittles. Okay? Inside supravital stains, we have a stain called new methylene blue. Okay? New methylene blue is used most of the time, like 99% of the time, to stain reticulocytes. There's another one that's like Cressel green or something, but we're not even going to talk about it because nobody uses it. All right? New methylene blue is a supravital stain used to stain reticulocytes. Okay? So, reticulocytes, what makes them reticulocytes in that polychromatophilic color is they actually have residual RNA left. Okay? So what you're going to see, and I'm going to show you pictures of these later on, is you've got red cells that are larger. That's one of the things you need to know. They're usually macrocytes. Okay? So we haven't talked about that yet. I made mention of it. Red cells are described as microcytes, normocytes, and macrocytes. Okay? Depending on their size. And how do we know their size? We look at an indice called MCV, mean corpuscular volume. And we're going to talk about that later on. It's not that important today. But reticulocytes tend to be macrocytes. They tend to be a little larger. All right? And the reason why is because they're not mature yet. Okay? One of the things you're going to see, size decreases as they mature. Okay? In all cells. We talked a little bit about this in the white blood cells, but the larger the size, the more immature the cell is, usually. Okay? So they go from large to small based on the maturity. More mature, smaller cell. Okay? But reticulocytes are not mature yet. They still have some characteristics of being immature. The color, the polychromasia, those remnants of RNA that you'll see when you stain it with new methylene blue. It lets you know that that's an immature cell still, okay? Even though it doesn't have a nucleus, all right? So right here, from here to the reticulocyte, it loses, I used to spell loses. Loses, that's not right. Is that right? Mm-hmm. Loses its nucleus. Okay? So in a metarube, the metarubicyte still has a nucleus. This one's called orthochromic normoblast, isn't it? Metarubicyte. Is it the orthochromic normoblast? So it still has a nucleus. It's a little tiny pinotic or pycnotic, however you want to say the word. So in other words, the nucleus is dying, okay? But it eventually is expelled from this maturation to the reticulocyte. Reticulocytes are never going to have a nucleus, okay? If it has a little bit of a nucleus in it, it's still considered a metarubicyte. Does that make sense? Okay? If it doesn't, it will, I promise. Okay? All right, so reticulocytes are very important. All right, so what do we use reticulocytes for? Reticulocytes are used to help us look at the treatment for IDA, for iron deficiency anemia. Okay, people who have anemia, their reticulocyte count is easily increased. Okay, because their body's trying to kick out new cells early. Does that make sense? So normal range for reticulocyte count. So write this down somewhere on this side. We're going to go with 0.5% up to 2.5%. Okay? Now, this number may be as low as 1.5, depending on what your book is, but this is a good range to cover males and females. Okay? Because females tend to have a little higher than males, because you guys are more prone for anemia than we are. Okay? So know that 0.5 to 2.5%. All right, babies. All right, now this is an adult. All right, adults, babies can all be up to like 3 to 7%. Okay, babies have a lot higher. Okay, your book may say 3 to 5. And that may be what it is, and 3 to 5 would be fine. I wouldn't mark it wrong on you. Okay. All right. Questions about reticulocytes. And we'll talk a little bit more about them a little later on. I'll show you some pictures of them, those kind of things. Okay.
Say it again. What's reticulocyte used to?
Say it again.
The reticulocyte.
It's used to basically look at like how treatment's working for anemia. Okay. People that have anemia, they usually tend to have a little higher reticulocyte count. Then when you give them iron therapy, the reticulocyte count should come down. It should come down to normal. If it doesn't, it means they're not absorbing the iron like they should. And so they may have some other problem going on with them. Okay? All right. So let's talk about normoblastic maturation. So it occurs in an orderly, well-defined process. Okay? So here's the characteristics. You see a gradual decrease in cell size with progressive condensation of the nuclear chromatin. So essentially what that means, when it's the ruboblast or the pronormoblast, it's a very lacy, smooth nucleus, the chromatin, the color of it. And the older it gets, the more condensed it gets and starts getting darker and ridges in it. Matter of fact, the rubocyte looks like a brain. The nucleus actually looks like it's a brain. Got ridges and valleys in it that you can see very distinctly. Okay. Eventually expulsion of the pynotic or pycnotic, however how you want to say this. I was looking at it last night. I've always called it pycnotic, but I was looking at it last night and I about decided it's probably pronounced pynotic. Okay? And the K is probably silent. Like a knot on a log. Huh?
What does that mean?
Pynotic means it's dead. It's a dying nucleus. Okay? Sometimes we'll see pynotic nuclei in settings, in neutrophils sometimes, okay? Especially if the patient's having a real bad infection. So they're having like a leukemoid reaction, okay? All right, the cytoplasm of younger cells is deeply basophilic, okay? So let me go ahead and tell you guys, anytime you see the word basophilic, all right, it literally means base lover. That's what it means, okay? Basophilic means that they're going to love the blue dye, okay, the blue stain. Most time the blue stains are basic stains, all right, like pH, basic, okay? So basophilic means blue, okay? Purple, blue, that kind of thing, all right? Whereas you see acidophilic means they love the acid, and the acid stain's usually a pink color. So you're going to see more of a pinkish color the older it gets. And the reason it changes like that is because even in the ruboblast or the pronormoblast stage, hemoglobin production has begun. Okay? In other words, the genes for making hemoglobin, that cell, it's doing it. It's making hemoglobin. Problem is, with a light microscope and the stains we have, it's not enough to show up. So you see that purple, bluish purple basophilic stain. But the more the cell matures, the more hemoglobin is being produced, because eventually that's its job, right? To carry hemoglobin, to carry oxygen, those kind of things. And so eventually, the further we get down to the metarube, then of course it has produced enough hemoglobin that it changes the cytoplasm color to like a grayish or pink looking color. All right, questions about that, guys? All right, and I got pictures, okay? You know I always got pictures for you guys. So I'll show you the pictures in just a minute, and we'll talk about those couple of things, all right? So 6 morphologically defined stages, which is what we're talking about. Remember, the HSC, the BFU, and the CFU are undistinguishable. They all look the same. They're all going to look like tiny lymphocytes, okay? Even though they're really, really naive, immature cells. So when we start talking about the normoblastic maturation, then we have to start talking about the ruboblast, pronormoblast, all right? And I'm going to go ahead and tell you, according to your book, I'm old, all right? Because it says the ruboblast, prorubocyte nomenclature, which is the names, is old and out of date. And I thought, well, yes, I am. I'm old and out of date. I'm a dinosaur. Should have died off a long time ago, okay? So pronormoblast is what you guys really probably need to learn. That the second part of this, okay? But I won't put both of them up there because you're going to hear the other one out of my mouth because that's what I was taught years ago and that's what's ingrained in my head, okay? So we do want to be on the same page with each other as we go, okay? Now, listen, I will give you credit for both names. Like when we're having a lab practical or whatever, you will always get credit for both names, whichever one you put, okay? You just got to make sure you get the right name, all right? Okay? So normoblasts, this is what we were talking about earlier. They spend about 5 to 7 days proliferating. When we talk about proliferating, what does that mean? What does proliferate mean? Huh? Reproducing. Proliferation means reproducing. Rabbits are very proliferative animals, right? So cells, basically what we're talking about, is they spend 5 to 7 days proliferating, meaning dividing, making new cells, okay? One BFUE or CFUE can make up to like 50 or 60 red cells. Does that make sense? Okay, as they go. All right. Reticulocytes. Once they reach the reticulocyte stage, so we're talking about the nucleate cells. Once they reach reticulocyte, they mature at about 2 or 3 days in the bone marrow. And then the first 1 or 2 of these days are spent in the marrow, and then they get kicked out into the peripheral blood. Okay. They mature. Then, of course, mature circulating RBCs. We're looking at that 120 days like we talked about before. Okay. Questions about any of that, guys? Doing all right? Okay. All right. So, erythropoietin. So, only cytokine important in regulating the final stages of erythroid maturation. Okay. Androgens. Y'all know what androgens are? You know what that term is? Androgens. Anybody? Huh? Has been a long time, huh? Well, I'm going to say that my androgens are less than most of your androgens right now. Okay? Because I'm 46 and some of y'all are 12, you know? 19, 18, whatever some of y'all are. Androgens are your sex hormones. So things like estrogen, testosterone, FSH, those female hormones, those kind of things, okay? My testosterone level may not be as high as, say, Elijah. You know, Elijah, how old are you, like 20? 21. Okay, I'm sure it's not then, man. So those androgens do have play a role on erythropoietin production also. And if you notice, like, especially puberty, when puberty hits, what do boys do mostly at puberty? Well, let's don't be vulgar there, Kara. When they hit puberty, what happens to males mostly? Voice changes, they shoot up, they skinny up, their muscles form more, all that. All of those things that happen require more red cells. And the reason they do is because they need more oxygen, because they're growing so fast a lot of times. My son is 16, but he literally from like 13 to 16 grew 4 or 5 inches, 6 inches. And I mean, some nights it was like he'd go to bed and wake up the next morning, he was an inch taller, And I mean, literally. you could tell they changed that much just overnight. Well, if your body's changing that much, you have to have oxygen to do that. And so your body compensates by making more RBCs for them so it can have the oxygen. So androgens play a big role. See, I'm 45, 46 years old now. I don't need as much red blood cells. I need it because I'm fat and old. And you got to, get out of breath walking to the door, But my body doesn't compensate. It doesn't make enough, more RBCs just because I'm out of shape, okay? It does what I should do. All right. So this link, if you guys clicked on that, I don't know if you did or not, it should still work. It's that bone marrow aspiration video, okay? I'd put it in here because we were talking about the erythropoietic marrow. You can see that red marrow that they were pulling out, okay? That's the reason it's there. So you got a link to that if you want to, okay? All right, there are other hormones also that play a role. They're just not a major role. Thyroid hormone is a big one. People that have hypothyroidism or hyperthyroidism, sometimes even things that have hypopituitaryism, they call it, where the pituitary is not doing its job. Those things affect erythropoietin levels, high or low, depending on what's going on with the actual patient. Adrenocortical hormones, so things like epinephrine, norepinephrine, cortisol levels, these kind of things all play a role in that too. And then growth hormone, like we were talking about just now. When those kids are growing, they're going to produce more red blood cells, so erythropoietin level goes up also. Okay? Questions about that? It's just general. I don't care if you know all of those, but just be aware that there are some. All right. So let's talk about the rubroblast. Rubroblast is also known as a pronormoblast. Pronormoblast. Okay? Pronormoblast. Produces 8 to 32 mature RBCs, so one cell is going through mitosis, about what? 16 divisions. That would be about right, wouldn't it? Up to 16 divisions, I guess. It doesn't necessarily have to do that. Largest of the normoblast series, so about 20 to 25 micrometers in diameter, so very, very large. High nuclear to cytoplasmic ratio, so there's that ENC ratio that we talked a little bit about in the last unit, okay? So make sure you're aware of that, all right? So cytoplasm. Cytoplasm contains large numbers of ribosomes, okay? What do ribosomes do? We got to stop right here and make sure we don't know this. If we don't know this, we're in trouble, okay? What do ribosomes do? Say it again, babe.
They carry proteins.
They don't carry proteins, but they do have to do with proteins. What happens at ribosomes? What do ribosomes do? What's their main job? May I remember from biology? Come on, Candace, you spent a lot of time in the biology department. What do ribosomes do? You remember? That a girl. They synthesize proteins. They assemble them. Okay. That is the location where messenger RNA goes that has the message to make a protein, and then that messenger RNA. So transcription occurs in the nucleus, right? Where the messenger RNA is formed. Y'all remember that from biology? DNA makes messenger RNA makes proteins, right? Transcription, translation. Ribosomes, by definition, is the location of translation, okay? The assembling of proteins, all right? So that's where the messenger RNA goes in, and then it summons transfer RNA based off of the codons and anticodons. Y'all remember all that? Okay? If you don't, you will in the next chapter when we start talking about hemoglobin, because I go over it with you guys to make sure you understand what we're talking about. Okay? But right now, just know that it has lots of ribosomes. It has lots of ribosomes because what does it have to make a lot of? Protein. Which protein in particular there, Kara? Hemoglobin. Thank you. Just mentioned it. Hemoglobin. So hemoglobin is being produced at this point, okay? But it's just not enough to detect with the stain. In other words, the color of the cytoplasm hasn't changed. It's still going to be that deep basophilic blue-purple looking color, okay? There's always a pale area next to the nucleus where the Golgi apparatus is. And this is a new stain that you guys haven't ever heard, okay? It's known as a Romanowski stain, okay? Romanowski is like a pack of Skittles also, okay? And the Romanowski stain that we use in the lab is called a Wright stain. Wright stain is W-R-I-G-H-T. Wright stain. Okay, it's a guy's name. Okay. Romanowski stains are just a category. This guy discovered a whole bunch of different types of stains that do cell stains. They stain cells. And so he gets kind of the credit of creating a whole batch of them. But Wright, the guy Wright, came up with the one for hematology. Okay. I will tell you this. There's also, let's see if I can spell it right. You guys will learn this in this semester. Sometimes it's Wright's dash, and it's, hang on, I never can spell this. Gimza, I think is how it's spelled. Maybe E and I may be swapped, okay? But it's called Wright's Gimza stain, okay? Gimza is a parasitology stain. It's a parasite stain, and we use it for malaria. You guys haven't talked about malaria yet, but you will, okay? Malaria is a genus, a group of organisms in the genus Plasmodium. They're protozoans. But y'all guys will learn that this semester, okay? But a lot of hospitals now, you actually find malaria very rarely in parasitology. Malaria is usually diagnosed doing a CBC on a patient, and you find it on the diff or the morphology. Because it shows such bad anemia, you have to do a morphology on the slide because it reflexes it because of the abnormalities, and then that's usually where they find it. So what they decided to do, instead of having a separate Gimza stain, is they said, well, we'll just mix Wright and Gimza. That way malaria will show up when you're doing your morphologies. Okay. So a lot of times you'll see in the hospital it's actually a Wright Gimza stain. It's a concentrate or a compound stain, okay? All right, small amounts of hemoglobin are present, but like we were saying earlier, they're not visible with the light microscope. So the nucleus, so let's talk about the nucleus. It's usually very, very large, takes up about 80% of the whole cell, okay? So therefore that high NC ratio, so more nucleus than you have cytoplasm, okay? Stains a bluish purple. The chromatin is fine, okay? So it's got a lacy look to it, all right? Has coarser chromatin patterns than a white cell blast, and we hadn't really looked at a myeloblast yet, but I'll show you some of those a little later on. And usually contains 1 to 3 faint nucleoli. Okay, what's a nucleoli? Okay, that's plural. The singular is nucleolus. So what's the nucleolus? Y'all remember the nucleolus? Y'all remember that from biology? Okay. So in biology, you had your nucleus, and then you had the nucleolus inside the nucleus. The nucleolus' main job is to produce ribosomes. Okay. So we talked about there's lots of ribosomes in the cytoplasm of these cells. They had to come from somewhere, and so they have the nucleolus. All right. So let me go ahead and tell you this: immature cells in the blast form, which would be this guy, and white blood cells and all this, and whatever the next series is, the next maturation. So in ours, it'll be the prorubicite or the orthochromic, no, the basophilic normoblast, right? Is that what we said? Basophilic normoblast, or the promyelocyte of the white blood cell series, okay? They're still producing enough ribosomes and proteins because they can still do cell division. But eventually they go away. Okay? The more mature the cell is, the nucleolus disappears or the nucleoli disappear, and that way you know it's going toward that mature stage. And I'll point that out again later on, but that is what that's talking about. Okay? All right, questions about that? All right, so what is that? It's a lymphocyte, right? So it's definitely our regular old lymphocyte. And so I put it up there because this white blood cell is the closest you're going to see to these immature cells, all right? This is exactly what HSCs look like. They look very similar to, probably a little less cytoplasm, but they look very similar to a lymphocyte, okay? But I put this up there is because sometimes you will actually see nucleated red cells on a smear, and you've got to be able to recognize that that's not a lymphocyte, okay? All right, so look what we got. So purple-blue cytoplasm, see it? Okay? Now, compared to the lymphocyte, look how large it is. So that lymphocyte's about this big compared to that ruboblast. Okay? All right, look how smooth the chromatin is. Okay, there's no ridges, right? Look how dense the chromosome is on the lymphocyte. See those ridges? You can see the dark light areas. Okay, that's condensing. That's being pushed together. Okay? Does that make sense? Okay? So if I push my skin together, I get ridges, right? That's what's happening to your cell. Okay? Your cell is getting smaller because it's getting mature. So you're starting to condense the DNA on it, okay? Because the nucleus is getting smaller, right? But here, you don't have that, all right? Now, you have to look very, very close, but you see that little round area right there inside that nucleus? If you can't see it, look on the slide when you get your PowerPoint, okay? Pull that PowerPoint up and zoom up on it, okay? Those little circles, there's one there, and there's one there. Okay? And actually, I think there's one right there. Those are those nucleoli that we were talking about, the nucleolus. That's a dead giveaway that you have an immature cell. Okay? Does that make sense? All right? If you can't see it, make sure you pull up the PowerPoint and look at it. All right? Because you're going to see them again. Probably in your atlas, it probably shows them. You can probably see in your atlas the nucleoli. It's probably close enough that you can see it. All right? Then this is that doning that we were talking about. So this is where the Golgi apparatus is, and it's just not staining. It's not taking up that right stain. Okay? Questions about that? All right, listen to me. You're responsible for this now. This cell. You have to know what this cell is. So if I put a cell on your test, okay, you have to know what it is. You have to be able to call it. That's a ruboblast. That's a pronormoblast. Okay? Yes?
What are the little white dots?
What little white dots? These,
Yeah.
it's just places that are not staining.
Oh.
You know, it may be other organelles that are just not picking it up. Okay? That makes sense? All right. Questions about the ruboblast or the pronormoblast? Okay? I think I got another one. Yep. Got some more. Okay? These are them. Okay?
What's the
Well, these two.
oh.
These are more immature. These are actually white cells. This, all white cells. This is actually a white cell blast. This is a myeloblast.
No, I was talking about from the last slide. They look the exact same.
They do look exactly the same.
Are they the same?
They are the same. I mean, it's not the same slide, but they look the same.
Oh.
Because they all have the same characteristics. Shape may be a little bit different, but the characteristics are always the same. Just like what I gave you the little flow chart for WBCs.
So these
They may not read textbooks, but they're still going to be the same.
Is this the same cell as before?
Yes,
Okay.
this is still a ruboblast. Okay, dark cytoplasm, high nuclear cytoplasm ratio, lacy nucleus, right? And if you look real close, you can see nucleoli in those. Okay, there's one, there's one, there's one, there's one. Okay, those are nucleoli. All right, and then you got that little doning areas. where the Golgi apparatus is, right up next to the nucleus. Okay, right here. See where it don't stain purple?
Mhm.
That's known as like a dawning. Now the sun's coming up at dawn. And it's there because the Golgi apparatus doesn't stain. Doesn't take up the stain. And so they refer to it as a dawning effect. Okay? Questions about that? Now all these over here are white cells in different maturation, with the exception of this one. This is actually probably a lymph. It's probably an immature B cell. This is a red cell. This is a red cell. That's a red cell also. This right here is actually, these two are probably the Erythrocyte, and that's a metaroot. Well, we'll talk about in just a minute, okay? But right now, this is two I want you to know. But, listen to me very closely. No more than you know right now. If you saw that on a blood smear, what would your brain should automatically say? That's not what? Normal. Right? Remember me telling you that? That's the whole point of hematology, is knowing what's normal and being able to recognize abnormal. You don't have to figure out what's abnormal, what it is. You just got to recognize that it's abnormal. And then we take it to that old $450,000 a year dude.
And those other two are
Him take care of
normal
it.
up there?
Huh?
So what are those?
These?
No, the pink one. The pink ones are
This just red cells.
Okay.
Yeah, these are just normal red cells. Yeah, normal red cell, normal red cell, normal red cell.
And that red cell down there.
This
what's-
guy?
Yeah, what's in the middle of that?
He just messed up. That's probably stain. You see all this trash in the background?
Yeah.
So it's like stain, precipitate, and those kind of things. And you'll see that a lot of times. Okay? But this is white cell, this is red cell. See the difference? And the colors? And they're always different in color-wise. Okay? And that's one of the things we'll talk about when we get to the white blood cell stuff. But by then, we've looked at the red cell stuff enough to know that it's always got that darker cytoplasm, different looking nucleus, those kind of things. Okay? Alright? Whereas, see, you can even see the nucleus here on this blast. This is a myeloblast. But you can see how it's a little rougher chromatin than that smooth, lacy chromatin of the red cell. Okay? And the cytoplasm color is different, usually, too. Okay, maybe a little bit more blue than that, but it's not going to be that bluish purple like a red cell. Okay? Questions about that? Ruboblast, pronormoblast, same cell. Different name, same cell. Okay? You need to learn pronormoblast. Okay? Pronormoblast. All right. Next one. Erythrocyte, which is also known as a basophilic normoblast. Yeah, basophilic normoblast is our next one. Erythrocyte, basophilic normoblast. Okay, smaller. That's the key to this. This one has very similar structures as the ruboblast, pronormoblast, except the size. Okay, it's still actively dividing. Cytoplasm is a deeply basophilic due to increased ribosomes, still the same. It has a perinuclear halo around the nucleus, okay, so it kind of goes all the way around. This corresponds to the mitochondria that is still there. It doesn't stain with the right stain either. And then you're seeing late stages have more hemoglobin production, okay, causing it the cytoplasm to be a lighter blue hue or pink areas, okay. So what that means is, remember we talked about this guy, the ruboblast, the pronormoblast, is dividing, okay? And even if it's dividing, though, these guys are maturing toward the next maturation, or to the next cell that we look at, all right? It's just like your kids. Your kids were born, when they were born, they were like this, and they were basically little blobs, all right? But every day they changed. They grew every day. All right? So even though this cell right here can still go through cell division, it's still going to be trying to go toward that maturation period and grow. Okay? So as it's growing, it's making hemoglobin, making protein. So the closer it gets from, essentially what it's saying is, the closer it gets from the prorubicite or the basophilic normoblast to the rubicite stage, which is the next one, you're going to see a change in its hemoglobin or its cytoplasm color. Does that make sense? All right. Nucleus NC ratio is decreasing now. You're beginning to see you're going to see more cytoplasm at this point. Nucleus is going to get a little smaller. Chromatin begins to get a little coarser, so you're going to start seeing some little dark edges or dark areas inside it. And at this point, usually the nuclei are not present, okay? If you do see it, there's usually probably just one, where there'd be up 3 to 5 in the pronormoblast, okay? Does that make sense? So the nucleoli is starting to leave at this, or start to dissolve, and it's usually not apparent in these, okay? And this is what one looks like, okay? This is a prorubicite, or a basophilic normoblast, all right? So in this case, you're still seeing the little dolmen. But look how the nucleus is getting smaller. So now we're seeing more cytoplasm than we did in that blast, okay? Still going to have that little area that's not staining. And then look at the nucleus itself. We're starting to see some dark areas inside the nucleus. So you're starting to get some crumping, pushing up against each other. And then there's our lymphocyte again for comparison. Okay? Questions about that cell? All right, same here. Okay? Now this one right here, I put this one up there for illustration. If I was calling this cell, this one I would call the prorubicite, okay, the basophilic normoblast, because the nucleus has gotten smaller. Even though the cell is larger, we're looking at that NC ratio, okay? And in this case, we've got more cytoplasm around than we do nucleus. Also, the nucleus is beginning to crumple up. I'm seeing some black in it compared to like this guy. See, this guy, we still got a huge nucleus, still seeing the nucleoli inside it, and you're seeing that dark cytoplasm around it. So if it was me personally counting this, I would call this the pronormoblast, and I would call this one and probably this one prorubicites or basophilic normoblast. Does that make sense? You see the kind of the difference in the cells? Okay. What's this cell right here doing? That's dividing. That's active mitosis. And you see this a lot of times on bone marrow slides. All these are bone marrow slides that we're looking at. Okay? You'll see this a lot of times because your cells are constantly going through mitosis. And sometimes you'll catch them in different stages of mitosis as you go.
The top one in the left-hand corner?
Top left. This one?
Yeah.
Yeah, I would
Is
call
it
that probably
still-
I would call this is a light cell. So this is a neutrophil. See the granules?
Yeah.
Gray. This guy I'd probably call, looking at it, I'd probably call it a metamyelocyte. The kidney-shaped one.
Oh, okay.
Yeah, this is a metamyelocyte, neutrophilic metamyelocyte. But look right here. What about that cell? This big orange granules? It's an EO. In this case, though, it's still got one big nucleus, so this would be your eosinophilic myelocyte that we talked about. Because that's the deal you got to learn. We hadn't talked about it a lot, but you got to learn blast, pro. You can't tell what the cell is without flow cytometry. But once they reach the myelocyte stage, you can tell if it's an EO, baso, or neutrophil based off of the granules and the cytoplasm. Okay, and that's a big thing students have a hard time with is they don't. We say myelocyte, metamyelocyte, and all that, and they just assume that it's always a neutrophil because that's our largest kind of cells that we have. But you can have myelocyte, metamyelocyte, even a band and a seg, eosinophil, basophil, depending on what's going on. They go through the same maturation process as the rest of them. Okay. And this right here is going to be our next cell. This is a more mature rubicide that we're going to talk about next as we go. Okay? Questions about that? Anybody? All right. So the rubicide is the next cell that we're going to make mention of. It is a lot smaller, so we're down to 12 to 15 micrometers in diameter. Cytoplasm at this point is usually more gray-blue because of hemoglobin production. Okay? At this point, there's lots of hemoglobin. Okay? Somewhere between about 80% of the hemoglobin that's formed is formed by the rubicide stage. Okay? So decreasing amounts of ribosomes, because we only need to make about 20% more to be complete. So we don't need quite as many ribosomes. And that's the reason their color's changing also, is because the number of ribosomes are decreasing. Okay? Nucleus, definitely a decrease in the NC ratio. Chromatin is very, very condensed, irregular. This is the one I was talking about, almost looks like a brain. You get good ridges and valleys in it. Okay? This is the last stage capable of mitosis. All right? Once we get to the metarube stage, the next stage, there is no more mitosis that is occurring. Okay? They have terminally going to terminate into what you have is what you have after that stage. Okay? All right? So what does a metarube or the rubicide look like? What is this one? Orthochromic? Is this orthochromic normoblast? Is a rubicide the orthochromic normoblast? Somebody got you say it. I'm sorry. Polychromatophilic normoblast. It's still a normoblast because it still has a nucleus. They still keep that consistent in the names. Okay? So polychromatophilic normoblast. Okay? Is the name you need to know it as. Alright, now look. This one could very easily be confused with a lymphocyte. Okay? It is actually about the same size. It's a little bit larger, but it is almost the same size as the lymphocyte. Okay? Now, this stain's not real good compared to this stain. Okay? But the characteristics are here. Alright? A lot of times the nucleus on a lymphocyte is not going to be perfectly round. Most of the time in the rubicide or the polychromatophilic normoblast, it is going to be almost perfectly round. They actually refer to this thing as a wagon wheel a lot of times. Okay? I don't see a wagon wheel, but I promise you that's one of the buzzwords that's used sometimes, is a wagon wheel nucleus. To me, it's a brain. It looks like a brain. It's got the grooves and valleys of a brain, and that's what I always teach my students. If you see that brain nucleus, go ahead and call it the rubicide stage at that point. Okay? Yes?
What color is it supposed to be?
It's gray. The coloring's about right. It's just the nucleus is not quite as dark as it needs to be.
Is it going to be that reddish?
No, that's what I'm saying. I think there's a problem with the stain in this particular picture. I think I've got another picture too in the next.
So what color is it supposed to be?
Gray, bluish gray. The cytoplasm.
The red part there?
The red part's the problem. It's usually, that's going to be blue too. It'll just be a darker blue. But it's the ripples and stuff. And that's what I was saying. I don't think that one stained as well as it needs to. This is more what you're going to see.
Oh.
This is more,
Okay.
and listen, okay, so we talked about stages of maturation. This one, 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 and this one would all be classified as rubicides or polychromatophilic normoblast. Okay? They're just in varying stages of their maturation. Okay? So like this guy here, how big he is, and this one, how big they are compared to these two. These two are about to go into, and you can see how the chromatin's getting really, really dark in the center here. That's getting toward pycnotic or pynotic that we were talking about earlier. Okay? So the next stage is the metarubicide, or what do they call it? Orthochromic normoblast. Okay? So the next stage is orthochromic normoblast or metarubicide. Now, at that point, the nucleus is almost completely pynotic. Okay? It's going to be like a black dot in it that's left. Okay? But see how the cytoplasm is starting to turn bluish gray? All right, see how this one does still have some blue in it, but by the time it gets here, it don't, and it's actually starting to turn a little pink, a little salmon color, okay? Once it reaches this stage, it's produced about 80% of its hemoglobin, okay? So it's getting that pink, the hemoglobin's picking up the stain at this point, okay? All right, so questions about that?
What was the name of this again?
This is polychromatophilic normoblast. Polychromatophilic normoblast, or the rubicite.
I have a question.
Okay.
If that's blue, what color is your shirt?
My shirt blue.
Do you see that those?
But what do you call that? Gray?
Purple.
Gray?
It looks purple.
That? This is purple. This is purplish. Yes, I'm with you on that. Purplish, yes. Yeah, I go with that. That's fine.
Okay.
It's just basophilic, what we call basophilic. Okay? But because it's starting to get darker in the center, you know that you're losing the nucleus. The nucleus is becoming dead. It's starting to die. Alright? But this is like, they call this like a gray-blue hue. And it's because of the hemoglobin production.
Is that the halo that you're talking
No, the halo's gone at this point.
Oh.
You're probably not ever going to see the halo no more. Because at this point, there's so much hemoglobin inside there, even though you couldn't see it. These cells can still carry oxygen at this point. You can see these in a normal peripheral smear on a person. So I shouldn't say that. I shouldn't say normal peripheral smear. You can see these on a peripheral smear of people that have severe anemia. Okay? Your body will kick these guys out of the bone marrow. This is like kicking the 12-year-old or the 13-year-old out and making them work.
Okay.
You know? They can work and they might can tote a few sticks and that kind of crap, but they're not sawing down the logs. You know what I mean? They're not running a chainsaw at 13, you know? So they can still tote the wood, but they can't do a good enough job. And so, but your body's in such desperate need, it just kicks them out early. Okay? All right, questions? All right, next one's metarubicite. Listen, when we get done with this, we're going to stop, okay? I know we're getting into an hour, but, you know. Metarubicite. So, cells 10 to 15 micrometers in diameters, very, very low nuclear cytoplasm ratio. Cytoplasm is predominantly pink or salmon color because at this point, most of the hemoglobin is created, you know, 80 to 85% of it. It still retains a tinge of blue because there are still some ribosomes available. Nucleus, really heavy chromatin, usually getting toward that pinotic stage. Can even be fragmented, okay? Usually it's eccentric. Remember what I told you guys about eccentric? What does eccentric mean? Pushed to the side. So it's almost going out the side, right? Or even partially extruded, because this is the last stage of the of having a nucleus. Okay, this one's also referred to as an orthochromic normoblast. Orthochromic normoblast is what you need to know it as. Okay, extruded. The nucleus is extruded while the cell's still in the erythroblastic island. Then it goes out a little bit away from that macrophage, and then gets expelled through the endothelial cells of the vessels. Okay, the expelled nucleus is then recycled by the bone marrow macrophages. So sometimes you can actually, when you do bone marrow aspiration, sometimes you'll pick up those macrophages, and sometimes you can even see the nucleoli inside those macrophages where they're in the process of digesting them. Okay, so definitely see red cell, but because it's so black and dark, it's known as pinotic or pycnotic. Okay, this is what you see all the time on a lot of patients that have anemia. Okay, it's usually this stage. This is like kicking the 17-year-old out and making them work and help you pay the bills. Okay, still not real good at working, but they definitely can hold down a job at that point. Okay, all right, no division on this one. All right. And I didn't put the lymphocyte up there, but you ought to be able to see that that's definitely not a lymphocyte. Right? Pink colored. Never see pink in a lymphocyte, do we? No. Okay. questions? These are extruded, getting almost extruded, very eccentric nuclei, I guess you'd say. Talking about both of them. All right. So you're responsible for these. Now at this point, you're responsible for all of these. All right. Reticulocyte, polychromasia, we talked about that. That's the next stage. So once the nucleus has been extruded, it is basically a reticulocyte at that point. Okay. It's usually a little bit larger. Okay. Talks about the cell being remodeled. We just pushed that nucleus out of the cell. So we've got to remodel the cell membrane to get it to fit or to close it off so that it can acquire that biconcave disc shape that a cell is. Okay. Cytoplasm is a blue tinge. This is known as polychromasia. These are actually called polychromatophilic RBCs or diffusely basophilic RBC. I've never seen that term used. Everybody always calls them polychromasia or polychromatophilic RBCs. There is a little bit of DNA left. Like we were talking about earlier, the remaining 20% of the hemoglobin is made during the reticulocyte stage. So that's important to know. Size 7 to 10 micrometers, so slightly larger, so they would be considered macrocytes. So if you're doing a morphology and you have lots of polychromasia, you say, I got 2+ polychromasia. You should also say you have 2+ macrocytes, because if you've got the polychromasia, you've got macrocytes. They go hand in hand with each other. Does that make sense?
Can you repeat that one more time?
Which part?
The two that go hand in hand.
Polychromasia, macrocyte. So what I'm saying is, if you have lots of polychromasia on your slide, in other words, cells that are bluish color, that are reticulocytes, then you should also have an increase in macrocytes. So your MCV number should be increased also as we go. Okay. We use supravital stains, new methylene blue or brilliant crescel blue, which nobody uses that. It's usually new methylene blue. The stain causes the precipitation of RNA and the mitochondria, and appear as punctuated purplish blue inclusions, and I'll show you pictures of that later on. There's a normal percentage, 0.5 to 2.5. We do an absolute concentration, can be calculated. Most of your instruments, that's what's really cool. You guys, I'm going to be mean to y'all, make you do a manual reticulocyte count so that you'll know how to do it, because you may end up in some podunk lab in Kodiak, Alaska or something, you know, somewhere up there. What's that? Life Below Zero. Where's that woman lives up there by herself in that place? Y'all know what I'm talking about? Y'all ever watch that show? Sue, ain't that her name? Is Sue her name? Y'all might be living up there with Sue, and you have to do a reticulocyte count on those Sue, you know? If you do, you're going to have to do it manually, because there ain't no machine up there. So I'm going to teach you how to do it. Luckily for y'all, though, most of your instruments nowadays do a reticulocyte count. Okay, they have a stain built into the machine, a little stain packet. It does exactly like you're going to do manually. It just does it on the machine. Where you guys will count like I think y'all count 1,000 cells, 1,000 red blood cells, and then you count the number of ticks out of 1,000 cells. It counts like 10,000 cells, you know, 100,000 cells.
We're gonna have
Right?
10,000?
No, you have to do 1,000 cells. No, it's not. It's not as much as you think it is. Okay, but you can do an absolute, and we look at absolute as basically a diagnostic tool or a prognosis tool to see how well iron therapy is doing for people that have IDA. Okay. So when reticulocytes are increased, you'll see polychromasia on the peripheral blood, like we talked about, also the macrophages. Okay. Some can contain small amounts of iron. Iron is identified by Pearl's Prussian blue stain. Okay. This is something that's very important. These two terms right here. All right. So if you use Prussian blue, okay, sometimes iron can, I don't know if I want to do this right now. I do want to do this part, okay? So sometimes you can see iron in a red cell, iron accumulates or accumulation of iron in a red blood cell on a rock stain. Okay, so like when you're doing a morphology, you may see these little speckles in it. Those are known as Pappenheimer bodies. Okay, you don't have to write that down right now. Okay, because we're going to talk about that in the CBC chapter. All right, but when they're stained with right stain, they have a different name than when they're stained with Prussian blue. Okay. Prussian blue is a iron stain, okay? So, if the cell is a mature erythrocyte, means it doesn't have a nucleus, and you stain it with iron stain, and you find iron in it, it is known as a siderocyte, okay? Siderocyte, okay? Everybody got that? Siderocyte, if it's a mature red cell, no nucleus, and you see iron in it, okay? If it is a nucleated RBC, no matter what stage it is, okay, so from the pronormoblast all the way to the orthochromic normoblast, okay, because it has a nucleus, and you stain it and it has iron in it, then it is known as a sideroblast. Okay? Alright? Now somewhere on this little note right here that you just made, write down the granules, okay? Iron granules are known as siderotic granules. Siderotic granules. Okay? Because Mr. Thomas is mean. I know y'all are shocked because I'm so nice most of the time to you guys. Okay? But I am really mean. So what I'll do a lot of times is I'll put Pappenheimer bodies up there or Prussian blue stain slide, and I'll ask you a question, and I'll say, what are the granules called? And then I'll ask you a question, what is the cell called? Does that make sense? And if you don't read the question right, you'll miss it. All right? So that's the reason I'm teaching you right now. Siderocytes are mature red cells that have iron granules in them, stained with Prussian blue. Okay? Now these guys right here, if it was right stained, they're known as Pappenheimer bodies. That'll come later on. All right? But if it's a nucleated red cell, no matter what stage, they're referred to sideroblast. Okay? And then the granules themselves are called siderotic granules. All right? Does that make sense? I know, I'm a mean guy. Okay? All right, so reticulocytes. I don't really like this smear, but it does show you that this is a larger cell. Okay? Y'all see that? All right? You can see it is a macrocyte compared to this. This is a normal cell. That one right there is a good normal red blood cell. You can see how much bigger that one is, okay? But to me, that one doesn't do real good with the polychromasia. So, I'm going to show you polychromasia. Maybe. Polychromasia. All right, that's a good one. So you see the difference in these cells? See how red these are? But then look how blue that one is. I got that blue tinge to it. That's polychromasia. Look how much larger it is compared to those guys also. Okay? That's what you see with the reticulocytes. That's polychromasia. Okay? This one is a good one. It's not quite as dark, but it is definitely a different color than the rest of them. So y'all see that? So that's what polychromasia is. That's a good one too. See how much difference that is in color compared to the normal red cells? That's polychromasia. That makes sense with you guys? You're like, okay, Mr. Thomas, we got the point. Okay. Making sure. Okay. And then erythrocyte we've talked a lot about already, so I'm not going to spend that much time, but you guys can read this. You guys have looked at red cells, so you know what red cells should look like. Okay? Little central pallor, about 5 to 7 micrometers in size. Shouldn't be almost. They shouldn't be sphere shaped. They should be round, but they shouldn't be look like a globe. Okay. They should always have that central pallor. Okay. Those are normal red cells right there. Okay. Questions about that? All right. We'll stop right there.